All right, in this video, we're doing an introduction to cell structure and organelles that you find within a cell. So we'll talk about different kinds of cells, like plant versus animal versus bacterial cells, the kinds of organelles that you would find in there, membrane structure, and we're going to start off talking about microscopy, just really briefly, because if you want to study cell structure, using a microscope is the only way you can realistically see a cell. They're just way too small for the most part. And the picture on the left is showing you a picture taken with a light microscope. And in this image, we're looking at salmonella bacteria that are so incredibly tiny, they're difficult to even make out. They basically just look like tiny little purple dots. And on the flip side, when you use an electron microscope that has many, many times more magnifying power, you can start to see the shape of individual cells, but they're still unbelievably tiny. And so a lot of the things that we know about cell structure are from microscopy and specific types of stains. And some things are from inference, because a lot of times we only get to see a two-dimensional structure of these things. And then you kind of have to imagine what they would look like in three-dimensional structure. So we use illustrations. To give you an idea of scale, this is a log-sized scale of different structures that you might find in cell biology. So a typical animal or plant cell is somewhere on the order of 10 to 100 micrometers. And that's an incredibly small value because remember, 1 micrometer is 1/1,000,000th of a meter. So these things are incredibly tiny. And then every click we go down on the scale, we're decreasing by a factor of 10. So bacteria are about the size of a human mitochondria. They're only about 1 micrometer. Viruses are on the order of nanometers. And then individual molecules and proteins are much, much lower, like down to single-digit nanometers. Anything below a virus, so these proteins and lipids and individual atoms, these things are far too small to see, even with an electron microscope. And so there are different methods for figuring out their structures, but we're only ever going to see those as illustrations or representations from a computer, because a scope just isn't ever going to be able to get that kind of resolution to visualize them. Right, so types of cells. The first major comparison to make is prokaryotes versus eukaryotes. And prokaryotes, you've probably heard before, means one of two things. We're either dealing with bacteria, which is most prokaryotes fit the bacteria category, or archaea. And archaea are much more rare. They're found in extreme environments like hydrothermal vents and sulfur springs, places where normal cells don't really inhabit. And prokaryotes are differentiated from eukaryotic cells because they don't have organelles. No mitochondria, no golgi, no ER. It really is just a cell with a cell membrane and a cell wall that form an outer barrier. The membrane is made of phospholipids, just like a eukaryotic cell is. And the cell wall is made out of something called peptidoglycan that makes like a rigid shell for the cell to live in. And then on the inside, in the cytoplasm, bacteria still have DNA, just like a eukaryotic cell would, but there's no nucleus. So the chromosome is just one big circular piece of DNA that's floating around in cytoplasm, and it's not really contained in anything else. The one thing that people often think of as organelles, ribosomes, those are found in bacteria, but remember, ribosomes aren't really true organelles. They don't have a membrane. They're basically just gigantic enzymes that read RNA and make protein, and all organisms have those. All right. In a eukaryotic cell, you get a lot more complexity. So here's where organelles start to show up. And remember, the definition of an organelle really just means a compartment within a cell. And so all of these different structures like a nucleus or endoplasmic reticulum or Golgi or lysosomes, all of these things are just membrane-bound compartments. And so it's almost like rooms within a house. We're sectioning these parts of the cell off so that they can perform specific functions. And the reason that it's helpful is that we can concentrate certain proteins and enzymes in specific locations. So you would expect to see very different enzymes and molecules in mitochondria compared to what you would find in Golgi or ER. That is what allows them to specialize. Just like if you went to a factory, you would find different machines and different equipment in different parts of the factory based on whatever is being made there. It's the same idea here. All right. And this lecture will focus mostly on the animal cell side of things. But remember, plant cells have organelles as well. And some of them are a little bit different. You have vacuoles that can control pressure inside the cell, chloroplasts that carry out photosynthesis. And otherwise, they're fairly similar, right? Still have ER, still have a nucleus, still have mitochondria, right? Those structures are all still present. We just add on a few plant-specific organelles as well. All right. Another issue that often comes up. The reason that cells are incredibly small and they can't be seen with the naked eye is that we have to give quite a bit of consideration to this idea of surface area to volume ratio. And the concept here is that as a cell gets larger, right, imagine this as being a tiny cell and then this as being a theoretically larger one. As a cell gets bigger, its surface area and its volume both go up, but the volume goes up by a second power, right, because it's squared, right, width times length, whereas volume goes up by a cube, right, because it's width times length times depth. And so if you go from a 1 millimeter squared cube up to a 2 millimeter squared cube, you get a dramatically larger increase in volume than you do in surface area. And the problem is when cells get larger and larger and larger, their volume gets so big and they don't have enough membrane to support the cell, right? Because the surface area is really like the cell membrane. And if the cell gets so enormous that it has a lot of cytoplasm, but not enough membrane for nutrients and waste products to move in and out of, it's not really survivable. And so that's why cells are kind of limited to that, 10 to 100 micron range for animal cells and plant cells. If they get too much bigger than that, they basically they outsize their volume relative to their surface area. All right. So speaking of membranes, because that's the surface area component, this is a mock-up of what a cell membrane looks like. And if you don't remember the structure of phospholipids and membranes, circle back to the playlist earlier on where we talk about lipid biological molecules. But the gist here is these phospholipids, remember, are composed of a phosphate head, right, that is hydrophilic and likes water. And it's composed of these hydrophobic fatty acid tails that don't like water, right? And then you have another phosphate head on the other side. And so these membranes spontaneously form because all of the hydrophilic parts of the molecule point themselves away from the membrane, because we would expect to find water over here outside the cell and also water in here inside the cell. And so if a molecule is going to diffuse or move through the membrane, right, if you imagine a molecule over here and we're asking the question, can it move through the membrane and get in here? The general rule is only molecules that are very small and hydrophobic can get through the membrane. Small because they have to wiggle through those phospholipids, and hydrophobic because there are fats on the inside. If this molecule is very hydrophilic and it likes water, it's going to be repelled by those fats and pushed back out. Okay, so because of this, we say that a cell membrane is a selective barrier because it's being very, very picky about what does and doesn't get to move in and out of the cell. All right, within the cell membrane, we get to add some proteins. So these protein channels that you're seeing, those are the selective part. If there's a molecule that can't move through, like pretend that our molecule out here is glucose. Glucose is a very water-soluble sugar. It can't go through the membrane all by itself. But if you add a glucose-specific channel and you open it, then glucose absolutely can move through the membrane, which is a good thing because now we can take nutrients into the cell. And so whatever that cell wants to allow in and out of itself, it has to put a channel or maybe an active transport pump in the membrane to make that happen, right? We also have things called glycoproteins and glycolipids. Those are exactly what they sound like. Glyco always means carbohydrate. So a glycoprotein is just a a protein that has glucose molecules or other carbohydrates attached to it. These are often used by the immune system as a sort of barcoding system to make sure that we can identify cells that really are human or whatever animal the cell happens to be in, and tell them apart from foreign cells like bacteria. All right, within the cell membrane, there are structures that we can form from it. So these microvilli are really just tiny extensions of the cell membrane. And if you zoom in close on a microscope, they look like almost like fins on a radiator. They're moving in and out and in and out, up and down. And their entire purpose is to increase surface area. So surface area meaning there's more membrane now on that side of the cell. So if this cell needs to add a lot of these membrane proteins, like for example, you see a lot of microvilli in the digestive tract because we need to absorb nutrients through protein channels. Those microvilli give you more membrane surface area so you can add more of those channels and transporters. That's what makes it easier to absorb those nutrients from your food and get them into the blood more quickly. All right, on to specific organelles. For right now, this is going to be pretty straightforward, and then later on in the playlist, we'll get to details about each of these. So right now, we're just covering the basics. The nucleus of a cell is the housing for genetic material. So that means DNA that's making up chromosomes lives in the nucleus, and in plant and animal cells, it never leaves. DNA is permanently inside the nucleus, except I suppose the nucleus technically breaks down during cell division, like meiosis and mitosis. But outside of that, your DNA stays in the nucleus, and when it wants to code for a protein, you convert that DNA into messenger RNA that can leave the cell and go out to be read by a ribosome. But the DNA and chromosomes themselves always stay trapped in the nucleus. And within the nucleus, there is even a subcompartment called the nucleolus. So that is a region within the nucleus that specializes in making ribosomes. And those ribosomes will also get exported from the cell. They're being shown here as these tiny little dots that are going to read that RNA made from your chromosomes and make proteins that that cell needs to survive. All right, so zoomed in a little bit further, this is what chromosomes would look like that you find in the nucleus. And they're not just pure DNA. Chromosomes are made out of something called chromatin. And chromatin is really just two things. It's DNA that's being represented by the purple strand in this picture. And it's a bunch of proteins called histones. And so when you wrap those DNA strands around a histone, that makes chromatin. And the entire purpose of the histones is they act as a way to organize the DNA and make so it doesn't just tangle on top of itself. And so it's kind of like, imagine wrapping up a length of string or Christmas lights or something like that. If you just ball them up in a coiled up blob, they're going to get tangled and tied in knots. Those histones let the DNA wrap themselves in an organized way so they don't get twisted. So when we need to wind and unwind them, it can happen in an efficient way. Okay, ribosomes. So this is what I was mentioning earlier. Ribosomes are made in a nucleolus, but they spend their life actually functioning out in the cytoplasm. And their entire purpose is protein synthesis. So in this picture, these purple blobs are your ribosomes. This pink strand of RNA, that is your mRNA or messenger RNA that I mentioned before. So when a cell wants to make a protein, it has to copy a sequence of DNA from a chromosome into messenger RNA, and that messenger RNA is what's read by a ribosome to make a protein. And so the mRNA is the code that's telling the ribosome what protein it needs to make, and then this chain of green shapes that's coming off of it, they're using that to represent the protein. So each of these little shapes is supposed to be one amino acid in the growing protein chain. That's what a ribosome does. Reads the mRNA sequence and then builds the appropriate protein based off of whatever that sequence coded for. The details of exactly how that works will come later on in the class when we start talking about the details of genetics. Right. Okay, mitochondria. Mitochondria are basically a power plant for a cell. This is energy conversion. And the idea is anything that you or an animal eats that has calories, whether that's glucose or other carbohydrates, or amino acids from protein, or fats, any of those three things can be converted into a form of energy that can enter mitochondria. And the reaction that takes place in mitochondria should release ATP on the other side. That ATP is effectively the currency for cellular energy. So any enzyme or protein that requires, like muscle contraction proteins that require an energy source, they're breaking down ATP. And once that ATP is used, it turns back into ADP. Right? ATP is just adenosine triphosphate. When you break that third phosphate off, you get ADP, diphosphate, and a free-floating phosphate all by itself. Those then go back to mitochondria, and you keep using energy from your calorie-containing nutrients to keep reattaching that phosphate. It's just a way to break these big energetic molecules down into smaller molecules like ATP that cells can make direct use out of. Okay, the next structure on our list is not technically really an organelle. It's not membrane bound, so kind of like a ribosome. These centrioles and centrosomes, the whole thing is a centrosome, and each one is made up of two centrioles. And these structures are critical for the cell when it comes time to divide. Notice them down here. Eventually in the class, when we get to cell division, you'll see that those centrosomes are the anchor point for microtubules that are going to attach to chromosomes during cell division. So when the cell has to divide and split, it needs to pull its chromosomes that it's copied to opposite sides of the cell. These guys are going to serve as a bit of an attachment or an anchor to latch those microtubules on so that we can separate chromosomes when the time is right. All right. Okay, chloroplasts. Chloroplasts in a lot of ways are kind of like reverse mitochondria. Mitochondria take in molecules with calories like glucose, and they give off carbon dioxide as a waste product. Chloroplasts are the opposite. They take in CO2 and energy from sunlight, and after the chloroplast's photosynthesis reaction is over, they're going to produce glucose as a product, right? That glucose then can either be used by a plant, or if an animal happens to eat that plant, the glucose is going to be converted back into CO2 and energy, and that CO2 can recycle right back around again to get reused. Okay, so same exact concept as mitochondria. This is a metabolic reaction that we've got going on. We're just running it in reverse by harnessing the energy of sunlight to convert low energy CO2 into high energy glucose. All right, ER and Golgi. So for context, notice our nucleus is up here, and ER comes in two varieties, rough and smooth ER. This picture is showing rough ER, and the term rough comes from the fact that when you put it under an electron microscope, it looks like there are little bumps all over the surface. Those bumps are ribosomes. So ER has ribosomes stuck to its surface, and they're actually making proteins and threading those proteins into the inside of the ER as they're made. And the purpose of this is the ER is kind of like an export, like factory for proteins. Once they're made, those proteins will get packaged into these little vesicles, which are like mini organelles, and get sent to the Golgi apparatus, which is a similar looking organelle that takes those proteins and it packages them into different transport vesicles that get shipped off to different parts of the cell. Some of them might go to the plasma membrane and the protein gets either secreted or incorporated into the membrane. Some of them might go to lysosomes or mitochondria or other parts of the cell. And so think of it like a shipping center where different vesicles that have different proteins get tagged and sent to different parts of the cell based off of whatever their function happens to be. Okay. This next set of structures, cytoskeletal proteins, these are proteins that you find inside of cells. So cytoskeletal, right? Cyto meaning cell, and skeleton, these are what give structure or framework to cells and sometimes extracellular proteins too. So they come in three varieties. Microtubules are, they got their name because they're like tubes in that they're hollow on the inside. And so tubulin is the protein, and each one of these individual circles that you're seeing, or spheres, is one tubulin protein. When you link thousands and millions of them together in a chain, you get microtubules. And microtubules are involved in cell transport. They're basically like railroad tracks for the cell to be able to move cargo up and down on. Right? Actin, in this image, each individual circle is one actin protein. And if you link those together, the resulting fibers, the whole thing, we call a microfilament. Actin is involved in movement and cell structure in a lot of ways. This is the protein fiber that muscles pull on to cause muscle contractions. It's also used to give structure to attach cell membranes to, so they're a little bit more rigid and they don't just flop around and burst as easily. Right? And in this picture up here, those actin filaments are highlighted in red. The microtubules are highlighted in green with fluorescent staining. Okay, and lastly, intermediate filaments. These are much thicker, stronger structural proteins that are used for any tissue that needs to resist a lot of stress. So as an example, keratin is an intermediate filament, and you see all these proteins that are sort of woven around each other very tightly. Think of them like ropes. Keratin is what hair and the epidermis of the skin and fingernails are made out of. Right? They're meant to withstand an enormous amount of stress and not tear easily. And so these tend to be extracellular, while actin and tubulin tend to be intracellular. All right. So this gives you a couple different ideas that are pulled together. So this is our cell membrane in this illustration. And notice that we've got some cytoskeletal proteins that are internal, right? Like microfilaments that are made of actin. Those collagen fibers that you see out here, those are similar to intermediate filaments, but they're made out of a protein called collagen, easy enough. They form something called the extracellular matrix. Extra meaning outside of. This is the network of proteins that forms outside of a cell. And the significance of it is these integrin proteins are embedded in the cell membrane, and their entire job is they link this extracellular matrix collagen protein with the cytoskeletal actin filaments, and it's basically acting like an anchor so that cells within a tissue can all be held together very tightly and not fall apart, right? Because now these protein fibers outside the cell and these protein fibers inside the cell are anchored together very tightly, and it gives a lot more structure. Okay. So to finish this off, the idea of cells being linked together, you have things called cell junctions, or you'll see them called intercellular junctions. And in this picture, each of these sheets right here is the membrane of one cell, right? So this would be one cell over here. This would be another cell over here. We're linking their membranes together so they don't fall apart, right? If you think of an organ like a liver or a spleen or a kidney, if every individual cell wasn't linked to the ones next door, they would just fall into a puddle. So these tight junctions are used when you want to make a very tight seal, basically a waterproof seal in between cells. The GI tract is a good example of where you would find these, because you want to make sure that the contents of the digestive tract don't just leak into your blood. And so each of these individual circles is representing a protein that almost acts like a zipper or a staple between these adjacent cells, so that if molecules up here try to squeak between the two cell membranes, they're blocked by those tight junctions. All right. Desmosomes are a different type of cell junction that doesn't create a perfectly strong seal, but they have an incredible amount of strength. And so you'll see these in tissues like the heart, where there's a lot of stress, the heart muscles contracting very hard, and we want to make sure that those cells don't get pried apart and separated from each other, right? So these cadherin proteins are basically linking to each other, right? Some of them extend from this cell and some of them extend from this cell, and they bind to each other very tightly in between the two to sort of rivet or staple the cell membranes together. Right. And lastly, gap junctions, still the same idea. They link cells together, but they have a pore in the middle so that ions and small molecules can diffuse through that pore and be shared from one cell to the next. And so if cells need to communicate with each other, especially in some muscle types like heart muscle, where electrical signals have to get passed from one cell to the next, and every time your heart contracts, there is a wave of ions that spread through all of your heart muscle cells, and they can share from one cell to the next by passing through those gap junctions. All right. Well, I hope this was a good introduction. We've got lots more detail to get into later on in the later chapters, and so follow the playlist, watch the videos in order. They'll build on each other, and eventually it'll all start to come together and make more sense.